All right, welcome back. So here's where we left off. So the liver, we can do full screen now. So the liver is required to maintain life. It is one of our most vital organs. However, it is capable of regeneration, and if 25 to 30% of the liver is healthy, it can regrow. So when a donor donates a portion of the liver, they're able to do that because both the portion the donor keeps will regenerate and the portion that the recipient receives will regenerate. If cancer spreads to the liver, life can only continue for a few weeks to months. Now, uh, when we talk about liver transplantation, um, it's generally preferable to do this from a cadaver, not from a living donor. It's possible to do it from a living donor, but it's preferable to do it from a cadaver because you can take the entire liver. And there's actually a high rate of failure with liver transplantation. It can be difficult to hook up all the blood vessels, even for skilled surgeons. So large portions of whatever is donated will die. And if you can just transplant the whole thing from a cadaver, then you've got a good shot at least some of the liver surviving. All right, so we also have extracorporeal liver assist devices, also known as ELAD. And this is kind of like dialysis, where blood is pumped through this device outside your body, and it performs some of the cleansing functions that the liver would normally do until a donor liver can become available. So ELAD is bioartificial, meaning it contains an artificial portion, the cartridges that have to be replaced, and a biological portion, which contains human liver cells that do the work of the liver for you. The patient's blood plasma gets passed through the ELAD device, where toxins are removed and liver secretions are added, and then the plasma is returned to the patient. Now, bile. Bile is a yellowish green liquid that the liver cells continually secrete. And bile is made up of water and bile salts. A major component are the bile salts. They're produced from cholesterol. All right, so they're modified cholesterol, and they help to emulsify fats in the digestive tract. Now, what does it mean to emulsify fats? Well, that means to suspend them in water. So if you have salad dressing, right? Not ranch or Caesar, but one of the more oily ones like vinaigrette. You'll see that it separates into water and herbs and then oil. And if you shake it up, you'll see little droplets of oil suspended in the water. Those oils or fats are emulsified, they are suspended in water, where normally um, water and oil don't mix. We're able to uh, suspend. It's a better word, suspend the fats in water by emulsifying them. All right. So bile salts are the only component that have a digestive function. So they act as fat emulsifying agents. Bile also contains the bile pigments, such as bilirubin and biliverdin, which are derived from hemoglobin breakdown. Remember, we break down hemoglobin into biliverdin and then remove the iron, and we get bilirubin. When a person has jaundice, the condition jaundice, where the skin is yellowing and the sclera of the eyes is yellowing, that's because there's an excess of bilirubin in the blood and it's getting deposited in the sclera or under the skin, and that's usually an indication of liver failure. Many babies are born with bilirubin. And I'm sorry, many babies are born with jaundice. And I've always heard it said that that's because the liver is immature. I got to thinking recently, because we just had a baby a week or so ago who had jaundice. Got to thinking if that might also be because of the turnover of fetal hemoglobin. Babies have their own special fetal hemoglobin that they stop producing at birth, and that has to get recycled. Not sure about that. Anyway, uh, also in the bile we find cholesterol and electrolytes. So hepatitis, remember hepato refers to the liver, is inflammation of the liver. And so there are many types, and it can be acute or chronic, and they will vary in severity. The most common cause of hepatitis is one of several types of viruses. Some people have symptoms and some do not, but they can be carriers of the virus. Some forms of hepatitis are bloodborne. Others can be transmitted by saliva. Yeah, contact with food, body fluids, objects contaminated with feces that contain the virus. Uh, right now, we're all worried about the coronavirus, and rightfully we should be. Uh, but you may remember a couple years ago, two now, we had an outbreak of hepatitis A in the area. Not nearly as serious as corona, but it was uh, uncomfortable to think about anyway. Hepatitis A is spread by in food that contains feces from someone who uh, has hepatitis A. And so what happened were there were a number of people at our fast food restaurants who were not washing their hands properly, and they passed hepatitis A on to customers, who then went to work at other restaurants and continued to spread it. All right, so antibiotics are not effective against viral hepatitis because antibiotics only affect bacteria. Antibiotics do not affect viruses. In fact, we do have some antiviral drugs, but generally they're only effective if taken within the first day or two. And usually you don't know you've been infected until several days after infection. Hepatitis C accounts for about half of all of the known cases of hepatitis. All right, so the gallbladder is this little muscular sac on the inferior side of the liver. In fact, it's kind of tucked away between the lobes of the liver here, the quadrate and the right lobe. It's sort of pear-shaped, and it's connected to the bile duct through the cystic duct. Okay, sorry, it's connected to the hepatic duct through the cystic duct. They join together to form the common bile duct. It's common because it carries bile from both the gallbladder and the liver. All right, we covered that. Gallstones. So the gallbladder normally concentrates bile salts, bile pigments, and cholesterol. Sometimes the cholesterol precipitates and forms solid crystals. When we talk about precipitation, in a chemical solution sense, that means that it falls out of the solution. Basically, it was suspended in a fluid and then it becomes sort of more solid and precipitates out. We say it just falls to the bottom. When that happens, it can form solid crystals, and these crystals can enlarge to become gallstones. All right, so excess bile concentration can cause gallstones. Too much cholesterol secretion by the liver can cause gallstones. Also, inflammation of the gallbladder. And you treat it through a surgery called cholecystectomy, which is where you remove the gallbladder. That's gallbladder removal surgery. Um, yeah. All right. So the hormone CCK or cholecystokinin causes the gallbladder to contract. In fact, the name cholecysto, all right, means gallbladder kinin moving, right? Cholecysto is another word for the gallbladder, or well, cholecysto refers to the gallbladder, and then kinin means to move. So this hormone's job is mainly to move or cause the contraction of the gallbladder. And in response, well, after contraction, bile is squeezed and moved down through the cystic duct into the common bile duct, down here through the hepatopancreatic ampulla into the duodenum. All right, and it happens when fats enter the duodenum. Chemoreceptors detect the presence of fats, and this causes the duodenum to release the hormone CCK, which circulates through the blood to the gallbladder, causing that contraction and then the movement of bile down into the duodenum. And remember, bile contains bile salts, which are responsible for emulsifying fats. So when we detect the presence of fats in the duodenum, we are going to ultimately release bile, which helps emulsify and suspend those fats. All right. So bile salts, we mentioned they're important in emulsification. Um, they also enhance the absorption of fatty acids and cholesterol by forming what are called micelles. Micelles are basically tiny fat droplets suspended in water. Um, they would not be visible to the naked eye. So when you shake up that vinaigrette and the oil and water appear to mix, what actually is happening is that the oil is being broken down into it was concentrated in a thick layer and now it's breaking down into smaller and smaller droplets of fat until you can't see them anymore because the fat's suspended in tiny droplets called micelles. All right. So the bile salts also help absorb the fat-soluble vitamins A, D, E, and K. And then almost all bile salts are going to be recycled. So gallbladder disease is characterized by pain in the upper right abdominal quadrant, as well as the back and right shoulder, sometimes causing nausea and sweating. You can observe the gallbladder through ultrasonography or ultrasound of the gallbladder, or an X-ray, which is called a cholecystogram. And gallstones can be found in the gallbladder as well as in the cystic duct, the hepatic ducts, or the common bile duct. You can treat it through cholecystectomy, which again is that surgery we mentioned to remove the gallbladder. You can also use endoscopy to find stones or remove them from certain areas, such as in the common bile duct. All right, so moving on, we've got the small intestine, which is a tubular organ, a muscular tube that extends from the pyloric sphincter at the stomach all the way down to the large intestine. And at the large intestine, we will have the ileocecal sphincter. All right. So it extends from the pyloric sphincter to the ileocecal sphincter. It fills most of the abdominal cavity, the small intestines do. They receive chyme from the stomach, as well as the liver and pancreatic secretions, bile and pancreatic juice. And it completes the digestion of the nutrients that are found in the chyme. Remember, carbohydrate digestion begins with salivary amylase. Protein digestion begins with pepsin in the stomach, and lipid digestion begins with gastric lipase in the stomach. That's where they begin, but they finish in the small intestine. The small intestine also absorbs the products of digestion. So when we're breaking down proteins into amino acids, those amino acids get absorbed in the small intestine. And then the small intestine will transport the remaining residue to the large intestine. So the small intestine has three parts, known as the duodenum, the jejunum, and the ileum. And the duodenum, the first part, is the shortest, and well, yeah, it's the shortest. It's where the majority of digestion occurs in the small intestine. The jejunum is the middle portion, which has a thicker wall, and it's more active than the ileum in absorption. And then the ileum is the third portion, the distal portion, the one closest to the small intestine. You could say it's the last portion we move through. And the absorption does occur here, but the majority of the nutrients will have been absorbed by the jejunum. Okay, and after the jejunum, there won't be a whole lot left to absorb in the ileum. Now, the ileum contains Peyer's patches, which you may remember from the lymph unit. These are lymph nodules, uh, And what they do is they capture bacteria that are trying to move from the large intestine up into the small intestine. Now, you probably know the large intestine is full of bacteria. There's relatively little, should be none really, ideally none, in the small intestine. Um, Now, how do we accomplish that? The bacteria are trying to replicate and move up. Well, the Peyer's patches capture them as they're trying to move from the large intestine to the small intestine. Now, why is it so important for us to prevent the movement of bacteria up into the small intestine? Well, because we went to all the trouble of hunting or farming or doing whatever we did to get our food. We went to all this trouble, and now we need to get as much out of it as we possibly can. We need to get as many nutrients out of it as we possibly can. And so the small intestine is relatively bacteria free. We are going to get first crack at all of the nutrients. All right, and then whatever's left over, we'll pass that on to the large intestine, and then the bacteria there in the large intestine can break it down. Sometimes they give us helpful things we need, sometimes they don't. Uh, but either way, we're passing on things we don't need to the large intestine. All right. If bacteria moved up into the small intestine, they would be competing with us for the food, and in fact, they would probably outcompete us. So anyway, the Peyer's patches, they're important. The jejunum and the ileum are suspended by a double-layered fold of the peritoneum. Remember, the peritoneum is the serous membrane that encloses the digestive organs. Um, and this double-layered fold is called mesentery. Now, unfortunately, we won't be doing any dissections in here, and honestly, even if we were meeting on campus, we wouldn't be dissecting the intestines. But in another class, maybe, if you've dissected a cat or a fetal pig, you would find that the small intestines are held together by this thin, transparent membrane that has to be destroyed in order to separate them out. That is the mesentery. If you watch any dissection videos, you'll see the mesentery surrounding the intestines. All right, so there's a double fold of peritoneum called the greater omentum, which drapes over the stomach, and it looks actually kind of like an apron, a big yellow apron that just kind of drapes over or lays over the intestines. The greater omentum, as well as the mesentery, are highly vascular, and their purpose is to support the digestive tract with blood vessels. Yeah. Here you can see some mesentery holding the small intestines together. All right. Now, as we move from the stomach, the first region of the small intestine we encounter is the duodenum. From the duodenum, which is very short, by the way, from the duodenum we move down here into the jejunum. All right, and then down through the jejunum into the ileum. And then finally from the ileum into the small intestine. Now, the last region of the small intestine is the ileum. The first region of the large intestine is the cecum. And so the sphincter, which separates them, is the ileocecal sphincter. Okay? The sphincter between the ileum and the cecum is the ileocecal sphincter. All right. So up here at the stomach, again, pyloric sphincter. Down here, ileocecal sphincter. Good. Just in this little cross section here, you can see the mesentery supplied with blood vessels. You've got small arteries, small veins, and then lymphatic vessels as well. Remember, in the small intestine, we have those lacteals. Those are small lymphatic capillaries whose job it is to uh, pick up dietary fats. All right, let's see. So in the frontal view here, you can see the greater omentum, and it literally just looks like a little apron hanging down. There are some large blood vessels here. There's also some fair bit of fat in the greater omentum. You can see it in a cross view over here, just kind of draped over the abdominal organs. All right, and again, that's a double fold of the peritoneum. And the peritoneum, again, is the serous membrane that surrounds the abdominal or the digestive organs. So here you can see it wrapped around the small intestines. You see this membrane wrapped around it. Okay. All right, so you've got the small intestines that are wrapped here. Right here is the transverse colon, so it surrounds the large intestine as well. And then on up to the stomach, and even part of the liver is surrounded here. All right. So many of the abdominal organs, the digestive organs, are enclosed in the serous membrane called the peritoneum. All right. So the intestinal wall has tiny projections of the mucosa called intestinal villi, which are going to increase the surface area for the absorption of digestive products. Each villus consists of simple columnar epithelium that has a core of connective tissue and then blood vessels and a lacteal, which is that lymphatic capillary that absorbs the fats, as well as nerve fibers. The free surfaces of the epithelial cells have microvilli, which are going to further increase the surface area for absorption. We're also going to have intestinal glands called the crypts of Lieberkühn, which are located at the bases of the villi, and they produce a number of products such as mucus as well as digestive enzymes. And then also, I've recently come to learn antimicrobial peptides. So basically antibiotics. They're pumping them out into the small intestine to keep bacteria out. There's a third structure known as the plicae circulares, and these are circular folds of the mucosa in the small intestine. And so here you can see a villus. This is a villus. A villus is a large structure, and it's lined with this simple columnar epithelia or pseudostratified columnar epithelia. And so we've got the villus, this long finger-like projection. All right, and then if you were to zoom into these cells, they have microvilli, which are more finger-like projections on their apical surface, all right, which is going to further increase the surface area for absorption, all right. Then there's a third structure called the plicae circulares. Where is it? Okay, just to go back, here are the microvilli again on each cell. So each cell has these microvilli, all right. And then we move here to these round structures called the plicae circularis. Now, it doesn't look round in cross section, but understand this would be like a ring that encircles the lining of the small intestine. And so each of these structures, the plicae circularis is lined with villi. Okay, you can see the villi here. All right, now we zoom in to the villi. You can see it's lined with the simple columnar epithelium. And then we zoom into the simple columnar epithelium, and you can see that it has microvilli. Okay, and so each of these structures, the plicae circularis, the villi, and the microvilli, are going to be increasing the surface area of the small intestine. And so even though the small intestine is only a couple meters long, there's actually enough surface area inside the small intestine, lining the small intestine, to cover a tennis court. All right, and again, we went to all the trouble of hunting or farming or whatever it is we did to get our food. And so we want to absorb absolutely everything from it that we can, and we do that by increasing the surface area of the small intestine. All right, so in addition to the mucus secreting goblet cells in the small intestine, there are specialized mucus secreting cells called Brunner's glands. And they secrete a thick alkaline mucus in response to certain stimuli. The intestinal glands secrete a watery fluid that picks up digestion products and then transports them into villi. These glands, this fluid contains no digestive enzymes. And then enzymes in the membrane of the microvilli include peptidases. Again, these are membrane-bound enzymes. The enzymes found in the pancreatic juice and in saliva and in the gastric juice, those are all freely floating around in whatever fluid, in their respective fluids. All right, now we have membrane-bound peptidases in the lining of the microvilli and the cells of the small intestine. We also have sucrase, maltase, and lactase, which are enzymes that break down disaccharides into monosaccharides. To be specific, sucrase breaks down sucrose, maltase breaks down maltose, and lactase breaks down lactose. The reason that most people, in fact 70% of adults, are lactose intolerant is because at age 5 we stop producing lactase, the enzyme that breaks down lactose into the two sugars that make it up, glucose and galactose. Let's see, lactase. I say 70% of adults are lactose intolerant to some degree. And that's because, again, as mammals, we're meant to drink milk from our mothers when we are very young. But we need to stop that and start eating solid foods at a certain point, right? So we stop producing lactase at about age 5. Except there's a mutation, and it's actually occurred twice in human history. It occurred once in Northern Europe, and it occurred again in Sub-Saharan Africa. The same mutation occurred in two different locations. Pretty interesting stuff. But those populations, or the descendants of those populations, are able to produce the lactase enzyme and are able to tolerate lactose. Good. Now, why do we need to break down disaccharides? Because remember, we can't absorb through the lining of the intestine. We can't absorb disaccharides. So like lactase, which is made up of a glucose and a galactose. I'm sorry, lactose, which is made up of a glucose and a galactose. We can't absorb that. We have to break that down into the individual sugars, glucose and galactose, and then we can absorb the individual sugars, the monosaccharides. Also embedded in the membrane of intestinal cells is lipase, and lipase is going to break down fatty acids. It's going to break triglycerides down into fats and glycerol. Sorry, finding it hard to talk today. All right, what else we got? All right, so you can regulate the small intestine's secretions, in a couple of ways. Mucus secretion occurs in response to mechanical stimulation or the presence of irritants such as gastric juice. Contact with chyme will stimulate the goblet cells to secrete mucus and the intestinal glands to secrete their watery fluid. Distension or stretching of the wall of the intestine will activate the nerve plexuses in the wall of the small intestine. And then distension will also stimulate the parasympathetic reflexes that trigger the release of intestinal enzymes. All right, so absorption in the small intestine, we mentioned the villi, the microvilli, and the plicae circularis all increase surface area for the small intestine. And the small intestine is where the vast majority of absorption occurs. We mentioned a little bit can occur in the stomach. Practically none, just enough that we have to say some can occur. A very small amount occurs in the large intestine. A very small amount. It's mostly water that gets absorbed. The small intestine, however, is where almost everything is absorbed. The vast majority of absorption occurs in the small intestine. In fact, it's so effective that very little absorbable material will reach the distal end, that is the ileum. Very little that could have been absorbed will reach the ileum. Carbohydrate digestion and absorption involves salivary and pancreatic amylase, which breaks down complex carbohydrates like starch and glycogen into disaccharides. We break them down into disaccharides, and then intestinal enzymes, that is, enzymes embedded in the membrane of intestinal cells, those will break the disaccharides down into monosaccharides. And as we just said, we cannot absorb disaccharides, we can only absorb monosaccharides, and so breaking the disaccharides down is important. All right, good. Here you see maltose. Maltose is made up of two glucose molecules, okay? And so the enzyme maltase breaks down maltose into two glucose molecules. And we can absorb the glucose, but we can't absorb the maltose. Maltose is too big. All right, so protein digestion. We mentioned pepsin in the stomach starts us off, breaking down proteins into small chains of amino acids called peptides. And then we have trypsin and chymotrypsin, which are going to break proteins and the peptides down into smaller peptides. And then again, in the lining of the intestine, we have peptidases. These are membrane-bound peptidases that break the peptides down into amino acids. Those amino acids are eventually going to be absorbed across the wall of the intestine through active transport and then moved into the blood vessels of the villi. And so here you can see a short peptide. Here we have one amino acid. Here we have another amino acid. Remember, for the amino acid, you have a nitrogenous head and then a carboxy tail. And so we're looking at one amino acid here, and then a second amino acid here. And for the upcoming exam, that won't matter so much, being able to recognize the chemical structure of an amino acid. But anyway, so I'm just pointing this out. You can see here the dipeptidase breaks down this short peptide into two amino acids, and now the amino acid can be absorbed. The peptide, the chain of amino acids, was too big, has to be broken down into the amino acids, individual amino acids. So fat digestion and absorption. Fats are emulsified by the bile salts, so they can be suspended in the watery environment of the intestines. Then the fats are digested by enzymes from the pancreas and the small intestine, such as the lipases. And then the fats are digested into glycerol and fatty acids by intestinal membrane-bound enzymes. And then the fatty acids and the glycerol are absorbed through a process involving several steps. They're absorbed into the blood or the lymphatic capillaries, the lacteals. Here you can see a triglyceride. You've got the glycerol molecule, and then you've got the fatty acid chains, fatty acid tails. They're long. This is an abbreviated form. And then lipase is going to break them apart. Here we have three fatty acid tails and then a glycerol molecule over here. Now, those fatty acids are going to diffuse into the intestinal epithelial cells, and then here they will join together to form structures called chylomicrons, and then the chylomicrons are transported across the basal surface of the epithelium and then into the lymphatic capillary, the lacteal, which transports this away from the intestine and then actually into the lymph and finally the venous blood. All right, just a second. All right, so moving on. So peristalsis, we've mentioned, that's the wave-like pushing movement that propels chyme down the small intestine. We mentioned it before when we were talking about the esophagus, but it's the same idea. It's a unidirectional movement of foodstuffs, right? So just as the esophagus uses those waves of contraction to push food down toward the stomach, the intestines will use peristalsis to move chyme down the length of the intestine. Segmentation is where we see these ring-like contractions that move the chyme back and forth. It's a mixing movement. This allows us to mix up basically digestive enzymes and chyme. Make sure that the enzymes get a chance to uh, work on all of the substrate. Parasympathetic impulses are going to stimulate both mixing and peristaltic movements, but sympathetic impulses inhibit them. Remember, parasympathetic is the rest and digest, so when it's time to rest and digest, the parasympathetic nervous system will be in charge. And when you see a saber-toothed tiger, the sympathetic impulses kick in, they stop digestion, and if you survive the saber-toothed tiger, you can digest your your chyme later. If you don't survive, then you don't need to worry about it. So if the small intestine becomes over-distended or irritated, you'll see a strong peristaltic rush that may sweep all of the contents quickly into the large intestine, and this causes diarrhea. All right, now we've already mentioned that the ileocecal sphincter joins the ileum to the cecum. The last portion of the small intestine is the ileum, first portion of the large intestine is the cecum, and the sphincter that joins them is the ileocecal sphincter. This is a capital I, this is a lowercase l. Ileocecal sphincter. All right, so the large intestine is called the large intestine because of its diameter, all right, because the size of the tube is larger. But it's actually much shorter than the small intestines are. The small intestines were quite long. Large intestine is only about 1.5 meters long. So what is that, about 5 feet roughly? Yeah, meters like almost 3.5 feet. So, all right, so about 5-ish feet long. At the distal end, the large intestine opens to the outside of the body through the anus. In the large intestine, we will see the absorption of some water as well as electrolytes. We will reabsorb and recycle the water and the digestive secretions. The large intestine also famously forms and stores feces. So the large intestine consists of the cecum, the first portion, the colon, the rectum, and the anal canal. So for the cecum, this is a pouch which forms the beginning of the large intestine. You'll see it written in a lot of places. This is a blind sac. All right. So the appendix is going to be attached to the cecum, and it contains lymphatic tissue. Do we have a picture? Yep. All right, so as we enter the large intestine, this blind pouch down here is known as the cecum. This tiny little finger-like, worm-like thing coming off of it, that's the appendix. And that's surprising to a lot of people to learn that this thing, which when it goes bad, uh, becomes very painful and can kill you, is so small. It's surprising to a lot of people to learn that the appendix, which can kill you if it becomes inflamed, is so small. But there it is, hanging off of the cecum. And the appendix has lymphatic tissue. When I was a kid, they told me, they told people, people said that the appendix was one of those organs that served no purpose. And that's not entirely true. The appendix does have a purpose. First of all, it has lymphatic tissue, so it's involved in sort of monitoring the composition of bacteria in the large intestine. It I would say that's probably its main role, is monitoring and adjusting the concentrations, well, the ratio of bacteria in the small intestine. That's a pretty important job. When it becomes infected and inflamed, however, it grows in size and it can rupture, releasing bacteria into the abdominal cavity, and that causes a condition called sepsis, which is frequently fatal. All right, what else do we have? You can live without your appendix, but uh, if you call that living, no, no, you can live without your appendix, but it does serve a purpose. All right, so moving back, the colon is the largest portion of the large intestine. You have the ascending, the transverse, the descending, and then finally the sigmoid portion of the colon. After the sigmoid portion of the colon, we move into the rectum, which extends from the sigmoid colon to the anal canal. And then the anal canal is the last 2.5 to 4 centimeters of the large intestine, and it opens to the outside world as the anus. In the anus, or the anal canal, you'll find internal and external anal sphincters, which guard the anus. And we'll talk about that when we get there. All right, so moving on, you have the cecum, then the ascending colon. Ascending means to go up, right? And so the ascending colon is this portion where the contents are moving up. You then have the transverse colon. Transverse means running across. And so we're running from the right to the left side in the the transverse colon. And then from here, we have the descending colon, where the colon descends or goes down. All right, and then lastly, you have this sort of curvy S-shaped portion. That's known as the sigmoid colon. All right, the Greek letter sigma is the equivalent of the Roman letter S. By the way, the alphabet that we use today is the Roman alphabet, or at least based on the Roman alphabet. So the Greek letter sigma is the equivalent of the Roman letter S, and so this curvy portion that sort of looks like a letter S is the sigmoid colon. Moving on from here, you have the rectum, which extends down to the anal canal. And then the anal canal is just the last couple centimeters here up before we reach the anus. All right, the anus is the opening to the outside world. All right, and so here we see well, on the left is a radiograph of the intestine. Doesn't quite look like the cartoon, but here you can see the cecum, the ascending colon, transverse colon, descending colon, then you see the sigmoid. All right, sigmoid's kind of clear, and then you move down into the rectum and the anal canal. Over here, you can see some rings of muscle. Okay, you see this internal ring of muscle and then these external rings of muscle. These are the internal and the external anal sphincters. And if I remember correctly, the internal anal sphincter is composed of smooth muscle, and it's under the control of the autonomic nervous system. And then the external anal sphincters will be made of skeletal muscle, and they are under your conscious control. All right. So the wall of the large intestine has the same 4 layers as the other tubular organs found in the alimentary canal. However, it will lack villi and plicae circularis, again because there's not a lot of absorption occurring here. You'll see longitudinal muscle is organized into 3 bands, known as the tenae coli, that run the entire length of the colon. And then the bands, they create pouches called haustra, which help form feces. And if we go back, you can see these sort of roundish structures here. These are the haustra, and they help form the feces. So the large intestine has little or no digestive function. It contains tubular glands that have goblet cells and secrete mucus, and that's the only significant secretion of the large intestine. Again, we're not absorbing a whole lot here. We're not breaking things down. Our goal is basically to form feces and move it out of the body, and mucus helps us do that. Mucus helps us move it along. We absorb water in the large intestine. About 90% of the water that enters the large intestine will be absorbed. We also absorb electrolytes here. Now, the large intestine houses intestinal flora. You'll also hear this referred to as the gut microbiome, and we're just now beginning to learn how important the gut microbiome is. In some of my own research, we've demonstrated that a high-fat diet causes inflammation, and that this is because the high-fat diet shifts the ratio of certain types of bacteria in the gut. Basically, you get more of one type, you got less of the other types, right? This leads to inflammation in a variety of ways. And it's interesting, by giving probiotic and restoring the natural fauna, the mice are able to eat a high-fat diet without experiencing the inflammation. Cool stuff. So, the flora, they break down contents that we can't digest, such as cellulose, and they produce the vitamins K, B12, thiamine. Now, just to be clear, we can't absorb the B12 that's produced in the large intestine. We can't absorb it. If we could, then we wouldn't need to find it in our diet, right? We would just be making it, or at least the bacteria would be making it for us in the large intestine. We do have to find it in our diet because we can't produce it ourselves, and the bacteria that do produce it in our large intestine, we can't absorb that. So the large intestine also, lastly, forms feces and carries out defecation. All right, so the movements of the large intestine are similar to those of the small intestine, but they're slower and less frequent. So the movements include mixing movements as well as peristalsis, about 2 to 3 times per day. The peristaltic waves produce strong mass movements, which usually follow meals. The defecation reflex relaxes the internal anal sphincter and then the external anal sphincter. Now, feces are composed of materials not digested or absorbed, and they also contain about water. Water is about 75% of feces. Now, remember, we do reabsorb 90% of the water that enters the small intestine, but 75% of what leaves is water. All right. So electrolytes are also found in feces. We find mucus, and of course there are bacteria as well as bile pigments that will affect the color. So the pungent odor is produced by bacterial compounds that include phenols, hydrogen sulfide, indoles, skatols, and ammonia. So disorders of the large intestine include diverticulosis, which is a weakening of the intestinal wall that leads to a protrusion of the mucous membrane, and it forms outpouchings that can become inflamed and infected. We also have inflammatory bowel disease, which is a broad group of disorders that includes ulcerative colitis, which affects the mucosa and the submucosa of the intestine, and it causes bloody diarrhea and cramps. We also have Crohn's disease, which is more serious and affects all layers of the intestinal wall, and it occurs in both the small and the large intestines. It can cause diarrhea and pain. And then we have colorectal cancer, which is a cancer of the large intestine or the rectum, and it's the fourth most common cancer in the U.S. Screening tests include fecal occult blood tests as well as colonoscopy. So lifespan changes to the digestive system are slow and slight, and they eventually include thinning of the tooth enamel. The teeth will become sensitive over time. The gums may recede. The teeth may loosen, break, or fall out. The GI tract becomes less efficient as we age. Peristalsis will slow, and this may lead to heartburn. If peristalsis slows down, then we are going to be sending the signal to the stomach that it needs to slow down its movement of chyme into the small intestine. We're telling the stomach to wait. And then if we are telling the stomach to wait, then the stomach will remain full, which increases pressure in the stomach and increases the likelihood that acid will move up into the esophagus. The gastric secretion will slow. Constipation may become more frequent. Nutrient absorption will decrease, and this includes the fat-soluble vitamins A, D, E, and K. You'll see the incidence of lactose intolerance increasing. You see a sharp spike in this after age 5. Um, yeah. The accessory organs will age, but typically not necessarily in ways that affect health. All right, so that is it for digestive. I will get back to you very soon with nutrition. Nutrition is a short, relatively easy lecture.